Thank you, Omri, for the invitation to submit the abstract to this summit. I'm trained as designer and architect and have been working at the intersection with agrobiotechnology, so it's quite a frightening experience for me to speak in this kind of place. So I'm going to show you the solar biobattery for use in bioelectronics. What is solar biobattery? Well, it's a thin film biophotovoltaic technology. We recently published, and it was featured in World Economic Forum not long ago, and really received enthusiasm from public, kind of scoring 130,000 Facebook viewings. But this is the technology which hasn't really come out of the laboratory for a long time, and using photosynthesis as an electron generator. Again, we received various enthusiasm from media outlets, varying fields, including printed electronics, to design, and to slightly more business-orientated interest into sustainable energy harvesting techniques. I've developed this technology in collaboration with a cross-disciplinary team involving electrochemist Andrea Fantuti, who's in this room today, and another biochemist, Peter Nixon's in this room as well, and Paolo Bombelli from Cambridge, who presented yesterday on his biophotovoltaic research. So what are cyanobacteria and algae? Well, you may know them as a third-generation biofuel source. My interest in this organism actually stems from the fact that they grow in various environments. And you see from the left sort of filamentous cyanobacteria growing on tree, and then another type growing in sand crust, a coccoid cells inhibiting the grain of stone, and there's also an amazing argo strain living in snow. So this diversity is extremely interesting when it comes to embedding the photosynthetic organism into a device. So my approach to the embedding of the cells was through inkjet method. I used thermal-inkjet, which is known to be very benign to cells. It's a technique used in cell engineering research. And the resolution of the inkjet I used is about 140 picoliter droplets, which is a little bit more precise and high resolution than the existing cell printer. For instance, the microvalve inkjet cell printer I tried had the resolution around 50 microliter. So in a way, to actually stick to thermal inkjet was to achieve a high resolution and also to have the flow between CAD to CAM. So just to extend a little bit more, the cell printer in general comes with its bespoke programming system. So therefore, to pattern anything in Vectorworks becomes extremely hard. So applying the existing inkjet technology used in ordinary office printing, etc., is actually a very scalable approach to cell printing, which hasn't been done with cyanobacteria before. So what happens to cells when they're printed? Since we need photosynthetic microorganisms to be alive and generating electricity through photosynthetic process, the cell viability becomes a very important criteria. Well, it turns out that almost 98% of the printed cells are very viable. And here you see printed cells grown for 3 days on paper. And one way to measure photosynthesis is to measure chlorophyll fluorescence. And we did that using imaging palm, and you can see that the photosynthetic activity is similar to that of cells grown in liquid culture. So this is to demonstrate that this new solid culture technique, the new way of growing algae through inkjet printing, cell deposition, and cultivation on paper, is actually as good as the traditional method of growing cells in liquid culture. For biophotovoltaic devices, there are two systems that exist today. One is the solid culture system that I'm presenting today, and the other is a liquid culture system, which Paolo showed yesterday. And the liquid system is slightly more predominant. Well, it's actually the most predominant system. And as you can see, the power output of the solid system is on par with the liquid culture, which is good news. So how does this work? The printed cells on printed conductor, it's based on the electrochemical cell. So you have anode and cathode, and photosynthetic biofilm deposits on the anode. And through photosynthesis, water molecule is split into proton, oxygen, and electrons, and the electron leaks through and travel through to cathode to trigger oxygen reduction, and this creates a voltage and pushes the electron from anode to cathode. So for the based on this system, we have a proof of concept, a prototype, as you can see, based on the zigzag pattern that we came up with based on some electrochemistry and insight. It's made of ordinary paper and carbon nanotubes printed. And the carbon nanotubes was a product commercially available. And printed cyanobacteria deposited on the anode. And the hydrogel, which is quite unusual to use in this system, is one made of polymer rather than agar or any other biological material, which hydrate the cells and acts as a proton membrane. So what we have as a prototype is extremely thin film system of biological solar panel. However, this system is interesting because, as you can see to your right, it retains the current production in the dark. So it is actually a battery conserving the energy from the photosynthetic reactions. So one way to improve the power output is to have a more efficient cathodic oxygen reduction. So we have moved on to a hybrid system where the cathode is no longer a printed carbon nanotubes on paper, but the platinized carbon cathode paper, which is quite conventional material to use in the BPV system or microbial fuel cells, but it's an expensive material. So suddenly the power output increases almost by 30-fold, and with this power output, we managed to produce overall current output of 1.5 to 2 microamp with voltage of 1.4 to 1.5, generating enough power to drive a digital clock. So other than the oxygen reduction parameter, there is this biocompatibility between cells and substrate that actually contributes to the power output, because this is directly related to cell viability and the health of cells. So to your left, you can see that the cells didn't quite grow on IT or PET. That is because of the lack of porosity of the material. So the cells still need some sort of supply of moisture and a media. So the paper has been an ideal substrate for this system. And what about the biocompatibility between cells and conductor? Yes, carbon-based ink have been known for a good interface with living organisms. But as you can see, that the chlorophyll fluorescent analysis of the printed cells on carbon nanotubes has not been very good compared to the cells just growing on paper. So this just shows that cells are actually happier growing on paper for long term. So this is also an insight into how we can, in the future, improve the longevity of this system. We are currently looking into different species of cyanobacteria that can best thrive in our semi-dry system. So in the top left, you see the sand crust glued together with desert cyanobacteria cells. They characteristically excrete polysaccharides, and they are actually very resilient in desiccation and under highlight. So over there you see the printed cyanobacteria growing on paper. And we have measured the electrocurrent output of this desert cyanobacteria in comparison to Synocystis, which is the strain we used in our prototype. And you can see that this strain inherently produces more photocurrent or current than Synocystis. So choosing the right strain not only improves the power output, but also improves the hopefully the longevity of the system by having some natural protective layer on their cells. So what brings us to ARM? We've been looking for feasible applications, and ARM has this great idea to power an ultra-low LOT circuits and CPUs with our biodegradable, environmentally friendly, and cost-effective energy harvesters, and hopefully for the use in biomedical or non-biomedical devices. In the big picture, there is this growing problem with e-waste. A breakthrough can pave the way for biodegradable electronics in the future, so that e-waste are naturally reduced over time and not being dumped illegally in countries like Ghana here. So I have 3 takeaways from this presentation. The solar biobattery technology is based on successful inkjet printing of cells using thermal-inkjet. And the parameters to be tested, including different species, biocompatibility, and cathodic oxygen protection. And this technology can in the future tackle e-waste. This research has been funded by EPSSC, IAA, and has launched a partnership since April with ARM. Thank you very much.
So we have time for questions, yes?
I believe you choose different patterns to print it out. So why did you choose a sawtooth and then digitize it?
You mean why we chose zigzag patterns, or
the sawtooth, the sawtooth shape of the printing cells. So have you tried different shapes like circular? Or other different patterns?
we have tried the circular pattern for the hybrid system. So we have tried, yes, another sort of pattern. But the zigzag pattern is based on actually Andrea's insight into ion mobility, so that trying to shorten the distance, traveling distance from anode to cathode, and hence contributing to an increase in the current output. But because we have been only operational at sort of proof of concept level, we haven't actually tested or explored optimized sort of more efficient circuitry patterns. So we would like to look into that in the future.
Because another question is that in terms of IoT, and I think these cells can provide enough power for powering IoT. But just another question is that I would be more interested in knowing the self-leakage, the self-discharge of that battery, which means when the battery is open circuit, and then what is the self-discharge rate of the cell when the IoT device is shut down? Because it concerns the long-term operation of the IoT devices, but it's somehow like engineering issues. It's not this stage. I'm quite interested to see future development or more investigation of this microcell.
Thank you for your question. Yes.
We will be looking into this. Yeah. So Anand and myself are involved in this project. Yeah.
So you showed like solid nanotube, but then you showed that it tended to kill off the bacteria that were over it. Did you try like grids or something, or like thin nanotube interspersed, with bacteria across them or something like that?
So the detrimental effect of carbon nanotube began to show around 30 days of growth, which is actually way beyond the electrocurrent production demonstrated by our reprinted system. So I forgot to actually highlight that the electrocurrent production was only demonstrated for over 100 hours. So in a way, the cell growth around 30 days is something that is actually, not, we're hoping that would be as good as the cells on printed.
What limits that to 100 days of current production?
Oh, sorry, 100 hours.
100 hours. What limits it to 100 hours? Do the cells just stop multiplying?
So we think it's the lack of the media supply. So as long as we actually replenish the cells with growth media, then actually the cell would continue to produce electrocurrents. But this is yet to be tested. Yeah.
Okay, so let's thank Marin again.